Hi, my name is Katia, and I'm currently working as a postdoctoral researcher at CIMBESTAB Unidad Irapuato. And today I will be talking about a meta-analysis of driver genes involving current diagnosis and tumor heterogeneity of glioblastoma multiforme. Glioblastoma multiforme is the most frequent and aggressive deadly primary brain tumor in adults with the lowest life expectancy, accounting for approximately 80% of all malignant gliomas. Although it can affect children, its incidence rises with age. GBM tumors are characterized by a remarkable genetic heterogeneity. Based on expression features, GBM tumors are classified into three subtypes: proneural, classical, and mesenchymal. In the left, a table where main features of tumor subtypes such as expression, histological, and patient prognosis are shown, highlighting that proneural has a better prognosis compared to more aggressive and proliferative ones, while in the right side, a model depicting parallels between stages in neurogenesis and glioma progression. GBM heterogeneity makes this type of cancer one of the most challenging to treat and consists of intertumor and intratumor feature variations. Intertumor heterogeneity refers to GBMs from different patients with altered and different genotypes and phenotypes related to diverse etiological and environmental factors. On the other hand, intratumor heterogeneity refers to the presence of multiple and different cell subpopulations within the same tumor, defining its topology and architecture. The comprehensive genomic classification of GBM paves the way for an improved understanding of tumor, which in the future may result in personalized therapy. Hence, there is an urgent need to further our knowledge of tumor heterogeneity, as it will help design better therapies against GBM and tumor recurrence. The current neuro-oncology practice has incorporated genes involving key molecular events that drive GBM tumorigenesis as biomarkers to guide diagnosis and design treatment. This study summarizes findings describing the significant heterogeneity of GBM at the transcriptional and genomic levels, emphasizing driver genes with clinical relevance. Based on a meta-analysis, 60 clinical reports were found from 2005 to 2020 that were fully test reviewed, using a combination of three terms: glioblastoma, clinical and case, and the BBS, Cochrane, Karger, and PubMed databases. Clinical reports were identified and selected by title and summary. All articles were evaluated using the guidelines of PRISMA. We selected and analyzed 18 driver genes that have shown the regulated behavior in patient samples. According to the biological process gene ontology, driver genes were clustered into three groups to determine their possible role in common pathways. In black notes, the first group includes driver genes related to DNA repair and involving chromatin remodeling pathways. Blue notes, the second group, include the cytoskeleton and cellular proliferation related genes, while red notes show tumor suppressor genes. Using bioinformatics pipelines and the TCGA database, we examined their mRNA expression in the different GBM molecular subtypes. First, we analyzed the overall profile expression pattern of each genes among GBM subtypes and the gene ontology classification. Then we analyzed the expression patterns of the driver genes clustered into three subgroups of patients according to their age: young, adult, and elderly patients. An important observation is that among tumors showing expression of this gene in patients under 30 years, the neural and mesenchymal subtypes were not observed. On the other hand, the driver gene expression in the mesenchymal subtype is only present in patients older than 80 years. Finally, to analyze the variation of these biomarkers at different stages of life in each subtype, we selected the genes with the most remarkable differential expression pattern, as we shown in this graph.
Summarizing, the gene expression analysis showed that the altered expression pattern for the 18 driver genes supports the GBM ontogenesis hypothesis from Selicu et al., which proposes that proneural subtypes can be generated from neural progenitors, and these cells may gain somatic mutations to become classical and consecutively mesenchymal subtypes. Our results also suggest that a combination of the herein presenting biomarkers could be a strong one to determine the GBM molecular subtype for patient prognosis.
Regarding the somatic mutations, we focus on analyzing the biological relevance of mutations using polymorphism phenotyping that helps to predict the functional significance of an allele replacement from its features by a naive Bayes classifier. The polyphen impact reported in the TCGA is a prediction of a mutation consequence being probably damaging, possible damaging, or benign. Driver gene mutations were clustered by patient age and analyzed by the protein phenotype impact values. Among the driver genes, the most affected among all patient samples were TP53 and EGFR. To better understand the behavior and mutations in patients, we analyze genes that are involved in GBM with the worst polyphen impact consequences. In left side, our results show that the young subgroup behaves differently as genes that are mutated are regulated by different transcription factors. Moreover, the transcription factors that regulate genes with mutations in the young subgroup share almost no transcription factors with adult and elderly ones. This might explain why the young subgroup has a divergent behavior in comparison with the others. On the bottom, the adult and elderly subgroups share most of the biological pathways while the young subgroup doesn't. In right side, in particular, we can notice that genes with mutation with a negative polyphen impact in the young subgroup share fewer identities with the elderly one. In summary, a pattern was identified fitting the stem cell model for GBM ontogenesis proposed by Selicu et al. In keeping with this, we found a combination of biomarkers with a strong potential to determine the GBM molecular subtype. A unique distribution of somatic mutations was found for the young and adult population, particularly for genes related to DNA repair and chromatin remodeling. Our results also reveal that highly lesioned genes undergo differential regulation with particular biological pathways for patient subgroups. We hope that this meta-analysis will help delineate future strategies related to the use of these molecular markers for clinical decision making in the medical routine.